Okay, now in part 2 we're going to review the actual movement of materials across the cell membrane and how it's accomplished. And you'll recognize a lot of the terms from 9th grade biology or 10th grade, I think it's 9th grade that you took it. Diffusion, osmosis, those kind of things. So kind of get yourself ready to review those things and get some key materials in your notes that'll contribute to the next lab we do. First, let's get diffusion into your notes. Now, write that diffusion follows the second law of thermodynamics, which governs all biological systems. That absolutely has been on an AP exam multiple times. Now, the second law states that all systems and the universe tend toward more disorder. We use the term entropy to describe that state. So make sure that enters into your notes. Also get in your notes that diffusion is the movement of a substance from an area of high concentration to an area of low concentration and requires no energy. It happens automatically. As you can see here in this slide, dye moves from an area of high concentration. So here you see the molecules of dye, high concentration of dye, none here. Can't be any higher or lower in this situation. So because there's such a high concentration of dye on the left, those molecules now will diffuse. They'll move or diffuse to the other side. And you see now here there's a membrane with breaks in it large enough for these molecules to fit through. So we're kind of assuming now that that semi-permeable membrane has openings in it that are large enough for these dye molecules to get through. So these dye molecules move from an area of high concentration to an area of low concentration. Now, how long will they move? They'll move until the concentrations are equal on both sides. Make sure you get that in your notes as well. We call that phenomenon equilibrium. So in any situation, whether we're talking about diffusion, which is particles, or osmosis, which is water, that will only occur until they're equal parts on either side of that membrane. And we call that system in equilibrium. Now, it's important for you to get in your notes that this simple diffusion requires no additional energy. Question for you. If it did, where would they get it? Where would it get the energy in an organism? If you said ATP or the ATP molecule, you'd be correct. We're going to be introducing that molecule again here shortly. Now, imagine opening a bottle of perfume in a room. The perfume molecules move into the room. Why? Yes, because the concentration of perfume molecules is greater in the bottle than it is in the room. So no energy is required. It just happens automatically because of the difference in concentration. Now write in your notes that the area where high and low concentration meet is called the concentration gradient. Now this slide now refers to it as passive transport. So write in your notes that passive transport is where no energy is needed. Now you also see here that they have diffusion and osmosis. These are essentially the same thing. Diffusion now is movement of particles like salt, sugar, etc. that are dissolved in a solution, whereas osmosis is the movement of the actual water molecules because of that concentration difference. Okay, and we'll talk about why, but you can clearly see that these molecules, these big ones, can't move through these spaces. So the only thing that can move are these little doojabbies here called water molecules. They can move freely between the two. So in this situation, we'd look at the concentration of water molecules and figure out where the highest concentration of water is, and it would move then from an area of high concentration to an area of low concentration of water. We call that process osmosis. Okay, let's move on to a concept called facilitated diffusion. Write in your notes that in facilitated diffusion, each transport protein is specific to the substances that will move. For example, the glucose transport protein in the liver, and you should get this in your notes, the glucose transport protein in the liver will carry glucose from the blood to the cytoplasm, but it won't carry fructose. So there's that specificity that we talked about. Also get in your notes that some transport proteins have a hydrophilic channel that certain molecules or ions can use as a tunnel through the membrane, which means they simply provide corridors allowing specific molecules or ions to cross the membrane. You see one right here. Here's a great example of a channel protein. So these molecules are clearly too big to get through the openings between these heads and tails. So in order for them to get there, they're going to have to use this channel now. And usually they call it the bouncer, because what do bouncers do? Bouncers decide what gets in and what gets out of whatever facility, normally a pub or lounge of some kind. But that's exactly what these protein channels do. They now facilitate the movement of molecules through the membrane that normally would not be able to for whatever reason. In this case, it's size. Size is a big determiner in terms of whether they get through there fairly easily. Now, here's the important part to remember about facilitated diffusion. It's still passive. Get that in your notes. Facilitated diffusion is still passive. It requires no energy. So this channel merely is, let's give a great example. Our little bridge over there, we couldn't get from one part of the building to the other without that bridge. So that bridge is merely a way for you to get from one side of the building over to the career center side. So it doesn't cost you anything, you just walk right across it. So there's no more or less energy required, but it gives you a way now to move from one side to the other. That's called facilitated diffusion, and there's no energy needed. So make sure you can explain that. So I also want to give you an example of a facilitated diffusion protein channel. They're called aqua, like water, AQUA, aquaporins. Okay? Water channel proteins are called aquaporins, and they facilitate massive amounts of diffusion. So make sure you get that in your notes. We'll talk a little bit more about them later in the podcast. But aquaporins, those are kind of a recent discovery. Okay, now what about extremely large molecules? How do they get through the membrane? Write in your notes that extremely large molecules are moved in and out of the cell membrane through vesicles and vacuoles. Vesicles and vacuoles, you see them right here. Through vesicles and vacuoles through two processes, endocytosis and exocytosis. So get those in your notes, and then we're going to explain them. So you can see here that endocytosis has phagocytosis and pinocytosis. And we're going to talk about those here very soon. So, two ways of doing it, endo and exocytosis. All right. Now, there are three forms of endocytosis. You see them here. Let's get each one into your notes and an explanation. First is a process called phagocytosis. So get that in your notes, phagocytosis. And this is where certain living cells called phagocytes ingest or engulf other cells or particles. Now, the phagocyte may be a free living one-celled organism, such as an amoeba, get that in your notes, or one of the body cells, such as a leukocyte, which is a white blood cell, get that in your notes. Now, in some forms of animal life, such as amoebas and sponges, phagocytosis is a means of feeding. Okay, so you see now here in a situation now where this is an amoeba, because here's a nucleus. You could tell this is a single-celled animal. And it's now actually using this phagocytosis to actually feed on whatever this is. Could be a plankton or who knows what it is. But whatever molecule it is, it's something it needs to feed on. In higher animals, phagocytosis, and we're going to get this in your notes, is chiefly a defensive reaction against infection and invasion of the body by a foreign substance. We call those foreign substances antigens. Okay? Antigens. So in your body, for example, your immune system uses phagocytosis as a defense mechanism. So this might be a bacteria or a virus that it doesn't recognize as yours. Remember the cell markers? And if it does that, it has these phagocytes that engulf and dissolve those foreign particles we call antigens. Okay, next is pinocytosis. Write in your notes that in the process of pinocytosis, the plasma membrane forms an invagination. All right, this is called a fold or invagination right here. So that's a word you may see on the exam. It forms an invagination, and you can see several of them right here. Forms an invagination or a pocket. Now, whatever substance is found within the area of the invagination is brought into the cell. In general, this material will be dissolved in water, and thus this process is also referred to as cellular drinking. Get that in your notes. I've seen that before too. Cellular drinking. So these are liquids now that it's engulfing with materials in it. Now this indicates that liquids and materials dissolved in the liquids are ingested by the cell. Now, it's different from phagocytosis in that this is a particle, just a solid. Now it's probably got liquid in it. But these are liquids now that have particles in them. So that differentiates phagocytosis from pinocytosis. Now, in general, this material will be dissolved in water, again, called cellular drinking. Now, lastly, is receptor-mediated endocytosis. Here's where you get the last one, receptor-mediated endocytosis. Now, write in your notes that receptor-mediated endocytosis is an endocytotic mechanism in which specific molecules are ingested into the cell. Now, the key word there is specific. All right, the specificity is not really there. For here, the specificity is absolutely there. Now, this specificity results from a receptor-ligand interaction. So receptors on the plasma membrane, you see them here, these little receptors on the plasma membrane, target tissue will specifically bind to ligands on the outside of the cell. That's why it's called receptor, here are the receptors, receptor-mediated endocytosis. So in this situation, these receptors are very specific to a particular molecule that needs to get in or out of the cell. That's what makes it receptor-mediated endocytosis. So make sure you can distinguish between those three things. So again, just a brief, I know this is going to be on the exam, just a brief review of the three. Phagocytosis is very non-specific, so is pinocytosis. So it's used for feeding in single-celled animals, but it's also used in your body as an immune response. Pinocytosis is now engulfing liquids with material in the liquids that it can be used or need to get rid of by the cell. And then receptor-mediated endocytosis, it has specificity in it. It's looking for a particular molecule and uses these receptors to find them. Okay, that's it for part 2. Take a picture of those notes, submit them to Moodle, grab that study guide, answer the questions, and I will see you in part 3. Bye.